Thank you, Mr. Hiroyuki Matsuda, for your heartwarming greetings. And ladies and gentlemen, we will now step to the next agenda, which is a report delivered by the person in charge of Japan Satraps Project, University of Tsukuba. To Dr. Seung-won Kang, the time is yours.
Okay, so thank you very much for joining today's JCC meeting. So first of all, just please let me share my slide. Okay. Okay. So let me start again. Okay. So can you, everyone? I hope you can see my slide. Okay. So and then I have still more than 5 minutes. So I think, yeah, I think I do not need to hurry actually. Okay. So just let me today. Just I'd like to introduce the how and what we are working on and how we have the how we have done in order to achieve our research goals during the last 2 years and during the last 1 year after the first JCC meeting. So we do hope that the second JCC meeting gonna be the good chance to discuss the previous research activities and just for the future research activities for the rest of 2 or 3 years in our research project. Okay, so as we as introduced by Dean, just our research project, the title is Breeding Innovation in Chili Pepper and Tomato to Accelerate Sustainable Vegetable Production in Tropical regions. So here, this is just I'd like to introduce how the project is organized and how our project is supported by from actually 2 agencies and Japanese agencies. One is JICA and the other is from the JST. So this JICA is actually belongs to the Ministry of Foreign Affairs in Japan, the Japanese government, and then JST, and then Japan Science and Technology Agency, and it belongs to the Ministry of Education, Culture, Sports, Science and Technology. We called the MEXT, was then MAP for the Ministry of the Foreign Affairs. So these 2 agencies, JICA and JST, from 2 government ministries, they support our Satraps project. So this is just brief the introduction of this how the project is supported from these 2 agencies, and just please Google Satraps. As you can see here, this is right, and then you can see some details what the Satraps project. So I'd like to introduce some research activities what we are working on in this Satraps project. So the first research activity is to develop the heat tolerance tomatoes. And then the second research activity is to develop the Anthracnose resistant chili pepper. So we introduce many several advanced breeding techniques. So in order to establish breeding platform for each tomato and chili pepper. So these are the research activity 1 and 2, and also not only to develop these new varieties of tomatoes and chili pepper, but also we're gonna analyze how our new varieties, the new cultivars we developed, will give on the impact on the agriculture industry in Indonesia. So we indeed, so that in this research activity 3, we're gonna analyze some economic in terms of this economic analysis and how we're gonna the cultivars gonna give some impact on the agriculture industry, so that we address the activities. So we're gonna analyze some economic validity of these two these horticultural crops, tomatoes and chili pepper. So the last research activity 4 is to establish Center of Advanced Breeding Technique. We call the CAPTEC or the CHAPTEC. So through this research center, we have the plan to distribute our outputs. So for example, these new cultivars of tomato cultivars and chili pepper cultivars to introduce to the farmers in Indonesia. Also, we will try to utilize our breeding platform we established through this project to other horticultural crops, for example, strawberries or the other vegetables, etc. And also, so therefore, this CAPTEC gonna be one of the core center, as we expected, and become this core center to develop new cultivars, varieties of the many vegetables in horticulture, the plants in the Indonesia too. And also we, not only for the scientific academia, but also we try to distribute our new varieties, new cultivars to the farmers and agriculture industry in Indonesia too. So the final goal is to accelerate, therefore the final goal is to accelerate the vegetable breeding process in tropical regions, not only in Indonesia, but also for the other countries in tropical regions in Southeast Asia, or also further to the Africa or the Middle Asia. So to promote the use of our new varieties and to establish the breeding system for the rapid development of new varieties in response to the rapid climate changes and demand for the production of these many horticultural crops. Okay, so this is just how we have been done. So this I made some timeline from starting from the June 22 until now. So this actually after the project is accepted from the from this JST. So we have started our preliminary project from this June 2022. So that during this time, from this June 22 to March 2023, it's almost 9 months or the 10 months. And then during this time, we have prepared and concluded many documents like this meaning minutes and the record of the discussion, and we also prepare the PO and PDM in order to conduct our project in December 5. And then second, we successfully concluded this CRA between our university and the Universitas Padjadjaran, and also this agreement between JICA and the UNPAD in the March 16, so that we could successfully. Actually, our project has started from the April 2023 in Japan side. And then after the Mr. Bingo, the coordinator of the project between Japan and Indonesia, and in Indonesia, Mr. Bingo has arrived on the November 15, so that the collaborative research has started as of the November 15, once after the Mr. Bingo has arrived. So that after that, we hold the kickoff meeting and also this symposium in the UNPAD on the December 4. So that here we have started actually the project between Japan side and Indonesian side. And then after 1 year, the end of this first year of this project in Indonesian side, we hold the first JCC meeting on December 4 last year, and then also at the time we have some huge progress, so that we have discussed Universitas Pajajaran, and then so that we planned the new plan for the construction of new building for our CapTech. And then this, so now we are here now. So this on today and December 1st, and then we now just holding the second JCC meeting. So this meeting gonna be more important to determine how we're gonna conduct our research project for the next 2 or 3 years. And also we are preparing for the midterm evaluation in Japan side. We're gonna hold this midterm presentation on the January 30 in 2026. So one of our activities in the CapTech is to extend our research network between the CapTech and other institutes. So we have discussed regarding this research network with JIRCAS, where actually Dr. Hoshikawa, the group leader of the first research activity to develop the new heat tolerance tomatoes. And also we have visited, and we are just working on how we can make some the research network between the CapTech in UNPAD and JIRCAS through the Dr. Hoshikawa. And also this tea park, and then just I belong to in my University of Tsukuba. So we are now just working on to make some MOU in order to make some research network between the tea park, just one of the research institute, Tsukuba Plant Innovation Research Center in our university. So this we are now just working on to make some research network between tea park and the CapTech in the UNPAD. Also, we have visited the World Vegetable Center in Taiwan. So if possible, we are also trying to make some research network between the UNPAD and the World Vegetable Center Taiwan. So we do hope we can visit the World Vegetable Center together in next period. So until these 2 years from this between the April 2023, actually we have the period that we have started our research project from the Japan until now. So we had already published the 4 articles, including review, and the 1 book chapter, published recently in last year. Also, this JST in Japan side is also supported to publish our chapter and the book to introduce our research project. And now, as long as I remember, just 1 article has been accepted, and just waiting for the publication, and 2 articles are under review, and more than 4 articles are in preparation. So this we do hope we can increase our output more in the next year. And also we have already 22 presentations in several conferences, in international conferences or the domestic conferences. So including 12 posters and 10 oral presentations. And also we heard about this 18 seminars and the workshops. So this is going to be, I think this would be a good quantitative outputs of our SATRES project. Also in terms of this capacity building, and we have accepted 2 long-term trainees in our University of Tsukuba. So these 2 trainees are working on their own research for as a PhD candidate. And also we have accepted 7 short-term trainees through these 2 years from the April 23 to until now. Yeah, so that we short-term programs of 2 months or these 3 months, so that actually there are 4 trainees in last year, and then 3 trainees for the short-term program in the first year. So we have already accepted 7 short-term trainees during the 2 years. Okay, so this is just all about our research activities during the last 2 years in brief, and thank you for your attention. Thank you very much.
Thank you very much, Dr. Sungwon Kang, for your report regarding this program. Continuing this momentum, it is now my distinct pleasure to present the main agenda of the Joint Coordinating Committee Satraps Universitas Pajajaran 2025. We will listen to the report on updates and reviews from each research activity of Satraps project. This session highlights the milestones, scientific progress, and collaborative achievements from our joint research between Indonesia and Japan. We sincerely appreciate the dedication of all researchers involved, and today we have the honor to hear updates directly from the leaders of each research output. The first research activity to be reported on the topic of development of heat-tolerant tomato will be delivered by Dr. Farida Damayanti. To Dr. Farida, the time is yours.
Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Thank you very much for the opportunity that is given to me to present the research progress from the research activity 1, breeding for heat-tolerant tomato. My name is Farida Damayanti. You may call me Farida. I'm in charge in this research activity 1. Now we move to the activities in this research activity 1. We have 3 different activities. The first one is development of near isogenic lines. The second one is development of tilling platform. And the third one is development of heat-tolerant tomato using genome editing techniques. And from those activities, we have some outputs, as you can see in this slide. Okay, for the first activity, development of near isogenic line population, we have 3 different sub-activities. The first one is evaluation of F1 and BC1 F1 generation, which include the evaluation of 6 population of F1 generation, and also evaluation of BC1 F1 generation as a part of background selection. And as the progress, we already have some plans for BC1 F1 that show bigger proportion of recurrent parent phenotypes, and those plans are used to develop the BC2 F1 generation. This is some documentation from this experiment. The F1 generation evaluation. In the left side is the leaf-related observation. We used a Nikon microscope that we got from this project and already established at the Captech laboratory. And in the right side is the fruit-related observation. Here we use tomato analysis software. And the second sub-activity is the development of BC2 F1 generation. We already crossed some BC1 F1 generation with their respective recurrent parental lines, and we already got 3 to 4 successful crossing from each parental line combination. And hopefully, if everything goes well, then we can harvest all the seed in late December or at least in January 2026. However, we face some challenges in this activity because we got quite high percentage of virus attack and also abnormality in flower development in beefsteak near isogenic line. And the third sub-activities is development of SSR marker for marker-assisted backcrossing, or MABC. We try to get the polymorphic SSR marker that we can use in MABC, and we also did some evaluation in the correlation between SSR marker that we evaluated with the heat-tolerant related traits. And in total, so far, we have 81 SSR marker that have been evaluated in 6 tomato varieties as the recurrent parent, the microtom as the wild type, and the mutant SLIA9-3 as the donor parent. We plan to have more SSR marker, around 25 SSR marker in further evaluation, because as you can see in this slide, that some part of the genome in the tomato, we are still missing the SSR marker. That's why then we try to get more SSR marker to be evaluated. And this is a small part of the result that we got also from this experiment. In the left side is the SSR marker visualization that related to flower and fruit traits. And in the right side is the correlation between SSR marker, some SSR marker with the flower and fruit phenotypes. Now we move to the second activity, the development of TILING platform. We have two different sub-activities. The first one is development of mutant population. In this second year, we already conducted the EMS treatment again in order to generate more M1 generation, and also we do the development M2 and M3 generation. Here, as the highlight right now, we have around 2,400 M2 plants from 200 M2 lines being evaluated at BRMP-TS Lembang. Here, the documentation from current mutant population development in 2025 cultivation. Right now, we have these plants at BRMP. And for the second sub-activity for development of TILING platform, we have characterization and evaluation of M1 to M3 generation. We conducted morphological and physiological characterization of M1 and M2 generation, and we also evaluate the M2 and M3 generation for heat-tolerant related traits, especially for the fruit set trait. And as the progress, the characterization and evaluation of M1 and M3 generation are in progress in BRMP-TS Lembang and also at Balai Tatanen in Universitas Padjadjaran. And this is some result that we got from this set of experiment. As you can see in this slide, we have some different phenotype or variation in mutant phenotypic in phenotype in M1 and M2 generation. And this is also some result from this experiment. As you can see in this slide, we already obtained suspected, because we should confirm furthermore, the tangerine mutant. As you can see here, we have orange color of flower and also fruit compared to the wild type here. And also we already got the curly leaf mutants and flower shape mutant and other phenotypic variation in M2 generation, as you can see in this slide. Now we move to the third activity, the gene editing for heat-tolerant trait. We try to establish the first sub-activity is establishment of a transformation system for Indonesian tomatoes. And our team tried to optimize the tissue culture procedure for Indonesian tomato. Here we use Intan for the first experiment, but if it's possible, then we also want to try another Indonesian cultivar. And the data analysis in this experiment are still in progress. This is some result that we got from this set of experiment. As you can see in this slide, we have kind of multiple shoot and callus formation, and also documentation of root induction in Intan cultivar. And the last experiment that we have as the sub-activity in this gene editing for heat tolerant, we tried to design guide RNA and the cloning. We chose two different genes. The first gene is hybrid proline-rich protein 1, and the other one is agamose-like 6. And for those two genes, we our team succeeds to design two guide RNA for each gene. And the validation of guide RNAs are still in progress. Meanwhile, for the Sanger sequencing, it's still in preparation at BRIN Cibinong. This is some result related to guide RNA design. And as you can see here in this slide, we already have 2 guide RNA for proline-rich protein 1. And also we did the preparation for Sanger sequencing for this gene. And our team already got 2 primer and validate the primer as you can see in this slide. For the publication, our team already did some oral presentation in international conference, as you may see in this slide, and also in domestic conference, and also as already mentioned by Dr. Kang that we already published the book chapter. And the last one is additional output for year 2 is the student engagement in the SATREP project. I would like to mention here because they are really important for us. Without them, we cannot do many things like this. In total, right now, we have around 15 students involved in the breeding for heat-tolerant tomato research activity. They are from the bachelor degree students, master degree students, and also doctoral degree students. I think that's all from me. I would like to acknowledge the SATREP project, GST, JICA, Universitas Pajajaran, University of Tsukuba, and also Ministry of Higher Education, Science and Technology for providing us the opportunity to work with this research. Thank you very much for your kind attention.
Thank you very much, Dr. Farida, for your report. Ladies and gentlemen, moving on to the next research activity, update and review on the topic of development of anthracnose-resistant pepper lines, which will be presented by Dr. Chitra Bhakti. To Dr. Chitra, the time is yours.
Okay, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Thank you very much for the opportunity. My name is Chitra Bhakti. I am the PIC of the research activity 2. In this moment, I will present about the progress of research activity 2, breeding for anthracnose-resistant chili pepper. As the background, chili pepper is one of the important crops in Indonesia. Most of the Indonesian dishes use chili pepper, and chilies contributed to inflation in Indonesia. Indonesian chili pepper demands reaches 1.7 million tons per year. Meanwhile, the production is around 3 to 3.5 million tons per year. The productivity of chili peppers in Indonesia is still lower than the yield potential, is around 6.84 tons per hectare. So the main cause of this problem is the plant disease. Anthracnose is caused by fungus Coletrotrichum species, which can cause significant losses to Indonesian chili pepper production. In general, farmers use contact and systemic pesticide intensively to control anthracnose disease, and that have a negative effect to environment. So using the resistant or tolerant cultivar is the most cost-effective strategy to control anthracnose disease in chili peppers. The promising output of research activity after 5 years anthracnose-resistant promising line of chili peppers are selected, registration of excellent line is prepared, and cultivation techniques are established and publication. So the list of activities that must to do for research activity 2, one, collecting chili pepper germplasm, two, developing a tilling platform using chili pepper, Tanjung 2, three, develop the selection technique for anthracnose resistant chili pepper, four, dissemination of Jajaran chili pepper cultivars, five, reaper registration for promising mutant lines of anthracnose resistant chili pepper, and six, establishment of cultivation technique for promising line of anthracnose resistant chili pepper. For the activity 1, there is sub-activity in research activity 2. Activity 1 is collecting chili pepper germplasm. The main targets are at least 5 local varieties collected from acreage in Indonesia, from North Sumatra, West Central and East Java, West and East Lombok, Jeneponto, and Endrekang in the end of the programs. For the results, 21 local varieties of chili from several regions in Indonesia have been collected. That is from South Sulawesi, from South Borneo, from West Java, from Jeneponto, from Lampung, from West Sumatra, from Aceh, and from North Sumatra. We also collected 31 genotypes of local varieties from Yogyakarta, West, East, and Central Java. We got from PIAT UGM. We also access the resiliency of this collection to the anthracnose pathogens. The results show almost of this local chili pepper accession exhibit susceptible and high susceptible to anthracnose. Only 2 genotype SLCB 195 and SLCB 596 exhibit moderate resistance during the green phase of fruit. We also collected 72 commercial varieties from the marketplace, consists of Capsicum annum, 12 genotypes, from Capsicum annum farfandulum, 14 genotypes, Capsicum fructescens, 31 genotypes, and Capsicum sinensis, 15 genotypes. So total collection of chili germplasm are 52 local varieties and 72 commercial varieties. This is the image of some local varieties collection, and this is the image of some collection from marketplace, and this is the image of some our collection. For activity 2, developing of chili pepper mutants line, we choose Tanjung 2 has the basic genotype for mutation because Tanjung 2 has the characteristics suitable for the tilling methods. Tanjung 2 has a compact plant architecture, medium tuck, beautiful rare fruit, can be harvested from 58 days and etc. So we develop M0 population from Tanjung 2, and after that we do the EMS treatment. Several experiment for the EMS treatment already conducted. We already conducted 4 sets of experiment to get lethal concentration of EMS. The lethal concentration 50 must be determined to ensure the high mutation frequency, but not too many plants die in M1 population. We applied the concentration of EMS 0, 0.5, 1.0, 1.5, and 2.0. With soaking duration is were 6 hours, 12 hours, and 24 hours. This is some of documentation, and at least the lethal concentration of EMS for Tanjung 2 is 1.99% and 0.82% for 12 hours and 24 hours soaking duration respectively. SC50 will be applied to create a big M1 population for tilling methods, and we also already collected M2 seed from 300 M1 plants. And for the preparing and planting M2 population, we already sowing 104 of M2 seed from 24 M1 plants since July 2025. The rate of M2 seed germination varied from 5.5% to 96%. Right now is 450 M2 plant in polybag in greenhouse at Lembang at phase generative stage, and phenotypic observation for M2 still on progress, and also optimizing procedure for DNA extraction for tilling method is still on progress. This is documentation. And for preparing and planting for the big population of M1 plants, we already 610 M0 sets were treated with EMS concentration 0.8%, soaking duration 24 hours, and planting in greenhouse at Ciparanje since July 2025. The rate of seed germination of 66.56%. And now there are 300 of M1 plants and 50 M0 plant in polybags in generative stage. Phenotypic observation is still on progress. Hopefully, we will harvest the seed in the end of the December or in the early of January. For the activity 3, develop the selection technique for the anthracnose chili pepper. We already sample of chili anthracnose pathogen have been collected from 6 provinces in Indonesia. Samples were collected from various chili plantation area in Indonesia. Because environmental condition and cultivation system will affect the type and virulence of the anthracnose pathogen. In total, 125 isolates have been collected from North Sumatra, West Java, East Java, Central Java, West Nusa Tenggara, and Sulawesi. And this is the result of the virulency level of Coletrotrichum. Most of the isolate from North Sumatra are high virulent, meanwhile the isolate from Central Java are moderately virulent. In the picture, the highest virulent of Coletrotrichum isolate will be used to screen chili mutant lines in order to identify candidate chili line resistant to anthracnose, maybe start from M3 generation of chili pepper. This is the training. We already got overseas non-degree training for 2 months, and we got one financial funding from Universitas Pajajaran and one financial funding for Ministry of Higher Education, Science and Technology. For the next year, we prepare some research proposal to get some funding, continuing activities 1, 2, 3, and find more space for activity 2. And find financial funding and space to start rejuvenation for 4 variety for activity 4, and also we prepare all genome sequencing for Tanjung 2 to support tilling method. That's all. Thank you very much for the attention. Thank you.
All right, thank you very much, Dr. Chitra, for your report. Ladies and gentlemen, the next research activity to be reported on the topic of economic analysis and impact assessment for new varieties will be delivered by Professor Eliana Wulandari. To Professor Wulan, the time is yours.
Okay, thank you so much. Please let me share my screen first. Well, good morning, ladies and gentlemen. Please, I would like to introduce myself first. My name is Eliana Wulandari. People usually call me Wulan here in Indonesia, but people from abroad usually call me with my first name, Eliana. But it's okay, you can call me anything, Eliana or Wulan. Yeah. Well, here we are from the research activity tree. From me as the PIC of this research activity tree, together with Dr. Zumi Saida and also with Professor Kawashima from Miyagi University. So for this project, we are responsible to conduct the research, especially about the economic analysis and also the impact assessments, yeah, especially for the applicability of the excellent lines of tomato and chili pepper. Well, as a broad project, we are in the output tree. We need two reports for the first indicator to the fourth indicator. And then for the specific activities, in the first activity, we need to conduct farmer economic survey. And then in the second activity, we conducted market survey. And now in the current year, we are conducting the activity 3, which is collecting farm management data. And for next year, we plan to conduct the consumers preference. Well, for the research activity 3 for the first year, which is in the 2023, we already conducted a farmer economic survey in the 4 districts, yeah, which well known as the production area of the tomato and the chili pepper as well. We came to interview the farmers in Cianjur, Ciamis, Bandung, and also Garut districts, yeah. And we found that in general, farmer growth 3 kinds of chili and also 2 kinds of the tomatoes. They have various characteristics in terms of the length, for instance, the taste, and also the color. And based on our interview with the farmers, especially for the chili, they also produce the various yield here regarding of the chili itself. And we also can see here, mostly of them use both the seeds from agricultural input kiosk, which nearby or surrounding of their house. And also they sold the heifers mostly to the middleman or the local trader, yeah, in their village. It also applies to the tomatoes, yeah, mostly because they are really depending on the middleman. So they also sold their harvest mostly to the middleman as well. And in the 2024, we conducted the market survey also in those 4 districts, yeah, to gain about how the trader or the local middleman, yeah, gain their harvest and also sell their products to the market. Yeah. And in this year, we also have opportunity to conduct a workshop, yeah, especially from Professor Shigekazu Kawashima, from our counterpart from Miyagi University, and also conducted a guest lecture, yeah, related to this research activity 3. And in this year, we also had opportunity to present our research, yeah, in the Seoul 2024 conference at Tsukuba, Japan. And also in this year, we also had publications, yeah, in a reputable international journal, which is indexed by the Scopus, yeah. Well, for the 2024, which is the market survey, we obtained some results that for the first product, which is the tomato, yeah, we can see the range of the purchase price per type of tomato based on the sources, which means the source of the the actor, whether the farmers as the one who produce the tomato, and also from the whole traders and also and the local market surrounding by the farmers itself. And with regard of the handling volume, yeah, per type of the tomato based on the sources again, yeah, we can see that, yeah, whether from the whole traders and the local markets, we can see they cannot, yeah, achieve the tomato from outside of the village. But again, as I mentioned before, that the farmers are really relying on the local middlemen or local traders in surrounding their village. And yeah, for the chili, yeah, it also applies the same with the tomato as well, which is the price varies that we can see from the slides, yeah, depending on the type of the chili they grew. This is also the handling volume. All right. In the 2025, we also had the second publications, also in the reputable international journal, which index in the Scopus. Also, as mentioned by the Dr. Kang, the publications of the book chapter also can be success in the very nice publisher, yeah, in the Springer. Well, in this year, we also had a very nice opportunity to conduct a training on economic and statistical analysis for market and household survey data at Tsukuba University as the host of this project. We did the presentations, yeah, for the SATERS project in Tsukuba University, and also we had a very nice discussions with experts, whether from the Tsukuba University and also the Miyagi University as the university for our counterpart working now. We also had the opportunity to visit the modern horticultural farming companies in Sendai. They are JR Fruit Parks and the Matsushima Sunfresh Tomato Company, and we learned a lot of, yeah, about the very nice technology growing tomato in Japan. And in this year, we also conducted farmers interview regarding of the farm management. Well, for the first thing about the farm management of tomato, we interview one of the, we call it with champion farmers, the farmers who is really, really good in growing, whether for the tomato and chili. Here for the, yeah, as we know that whether for the tomato and chili, those two products are strategic horticultural commodity with a very high economic value and good market potential, especially in Indonesia. And they need really intensive management, yeah, started from nursery stage to marketing, yeah, to achieve optimal productivity, yeah, started from seedling. Yeah. We found that, yeah, as the champion farmers, they grow, yeah, for the first thing by selecting superior seeds, yeah. And then for the seedling media, the farmers mentioned that this media must be sterile and also fertile, yeah, to support optimal seedling growth. For the land preparations, yeah, the first thing, this activity includes the weed removal, yeah, by using the cultivator, and also they applied basic fertilizations, yeah, such as manure and organic and other organic fertilizer. And then for the plant maintenance, yeah, the farmers are doing the watering and replantings, weeding and prunings, yeah. Also following fertilizations and also stakes installations, yeah. For harvesting, yeah, they do require the harvesting criteria, yeah, based on the fruit's ripeness. Yeah, we mentioned something like that. Yeah, and for the marketing, yeah, include selling through vegetable middlemen surrounding the productions area in their village, and also the successful tomato farming requires the integrated approach, starting from the selecting superior varieties, implementing appropriate cultivation technology, integrated pest and disease control to effective marketing strategies to improve farmer welfare and also meet the ever-growing market needs. And for second commodity, which is the chili pepper, it also applies the same with the tomato as well. Started from the seedling and then also the land preparations, maintenance, and also the harvesting. They started by selecting the quality of seeds, and then also for the seedling medium, they also use the medium that are sterile and also fertile to support the optimal seedling growth. And then they also applies for the land clearing and also drainage systems, yeah, and basic chemical fertilizer, yeah. And then for the maintenance, yeah, they do the activities including the watering, weeding, and also the pruning, yeah. In the harvesting, they also select the chili based on the color, the ripeness, and so on. And finally, for the marketing, yeah, they require or they determine the desired level of the ripeness, yeah, and then because they are really depending on the local middleman, so most of the farmers will sell their product to the middleman. But the nice thing is they don't have to transport it by themselves because the middleman will come to pick up the harvest to their field. So that's all that we can present. Thank you very much for your attention.
Thank you very much, Professor Wulan, for your report. After we have listened to several updates and reviews, now we will continue to the last session with the research activity topic, Establishment of the CAPTEC. This report will be delivered by Dr. Nono Karsono. To Dr. Nono, the time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ohayo gozaimasu. Selamat pagi, Bapak Ibu. I would like to present output for the establishment of the CAPTEC. The PI is me. My name is Nono, and the second PI is Dr. Vira, and thank you very much for the opportunity and also for Jika representative and Dr. Kang and what you call from GST in the Zoom meeting. Also thank you very much for your support. I would like to present this output 4. This output 4 is about the establishment of the CAPTEC. The activity, there are some activity, establish the CAPTEC and build research network, and also establish training programs, access the applicability to other vegetables, and hold workshop and seminars. And the last one is implement short-term and long-term programs. Output 4 is expected to establish and functioning to accelerate the CAPTEC for the vegetable breeding process and to promote dissemination of new cultivars. Some of this activity we try to what we call to fulfill. However, some of them we will be conducted until maybe 5 years, next 3 years, sorry. This is the concept of our ChapTex. Is a center of excellence with the focus on the improvement of crop variety and breeding method for the benefit of Indonesia and other tropical countries. And we have mission to be leading institute in development of crop variety and breeding platform in Indonesia and other tropical country. The mission is to develop excellent multidisciplinary plant breeding research and educate future plant breeders, because now in Indonesia, maybe in other countries, young generation is much less attractive to agriculture and especially for plant breeding studies. So we expect we can still educate plant breeders. And the third one is conserve plant genetic resources. As we can see from the project of chili pepper and also tomato, we create many mutant lines. The the fourth one is to provide excellent laboratory services and support sustainable crop production and establish collaboration. We do hope all of mission we can achieve within the projects execute. And the objective of the ChapTech is to produce new cultivars suit user needs and shorten the plant breeding process through technological innovation, and to accelerate the adoption of superior cultivar for vegetable crops that benefit to society, develop national international collaboration with various stakeholders. And we try to answer some questions regarding on the sustainability that we try to sustain by producing cultivar patent and plant variety protection, and we do the services, for example, DNA analysis, PCR analysis, crop testing, and others. And we do training and workshop for junior high school maybe or senior high school, and for the society, especially for farmers. And to establish sustainability, we try to find the research grants. That's all what we call the plan, our plan to be sustained. The first one, I would like to report that the decree of the ChapTex has been issued on April 29, I think, 2024, last year. The decree is from our rectors. This is the roadmap of the ChapTex development. As we can see here, we have initiation phase, short-term, mid-term, and long-term. Hopefully, we can reach our ChapTex by 2029 and 2033 by doing R&D for tropical crops and database availability and accessibility, and establish international collaboration. This is, I would like to report about last national seminar that we did. The topic is utilization of biodiversity in improving crops resistant to stress. We did in 11 July 2024 by online platform, and there are many speakers from Professor Kawashima, Dr. Ratli from BRIN, and also Keng Hoshikawa from JIRCAS. And we also did plenary session from Syngenta and from UNPAD and PT Bayer. It has been attended by around 220 participants and 70 presenters, and from lecturers, researchers, and also students. This also international seminar that we did last week, I think, on Friday, 28th. The topic is the Grand Challenge in plant biotechnology commercialization. Some speakers were invited, including Professor Azura, Dr. Satya Nugroho from BRIN, and also from Biotech Indonesia, Professor Herman, Michael Leaders from Bayer, and also Balasubramanian Ramaya from Corteva. They share their talk and experience regarding the commercialization in plant biotechnology products. It was attended by maybe around 232 participants and 42 presenters from lecturer, researcher, and students also. This is the human resource development that we already did in for some human resources that we have. We have training, long-term degree training, doctoral degree, Dr. Santika and Timazul Azmi, short-term non-degree training, Dr. Farida, Dr. Fiti and Faza, molecular biology analysis, and also Professor Ulan and Dr. Zumi did economic and statistical analysis for market and household survey, September, October 2005, and workshop also last year in October and July and October, and resource person in BIN. This is collaboration network that we have created between ex-Balitsa and also draft reference with the BRIN, because we already applied actually, but we have to revise because they changed their format for cooperation. And now we are under review. And we have opportunity also by visiting by Ambassador of Japan, May 15th, 2024. It was attended by our dean and some dean from other faculties. And so we we report some progress in our projects. This is the design of our Chaptech building that now we already renovate and fulfill by this equipment. Maybe after lunch we will visit it. We have some equipment from this project, from PCR and PP system, and many more that will be applied, will be used for our research in the Chaptech. This is new building of the Chaptech. We partnership with FRIES at Jatinangor Experimental Station, or we call Ciparanje. Maybe we do hope that it will be finished at the end of this year, and the key will be handed for us. So there are two center of excellence there, FRIES and the Chaptech. This one is a research grant funded to support Satra project, come from UNPAD and Ministry of Education and Research in 2024 and 2025, and we don't know actually for research grant for the next 2026 and 2027, 2028. Hopefully, maybe the same. Hopefully the same. This is, as already mentioned by some PI, and also from Dr. Kang also said that we already published some, this is the socialization of our Satra project activity actually. in the book from Springer, I think, Plant Genetic Resource and for Sustainable Crop Production. And our title is Development of New Vegetable Varieties for Sustainable Production in Tropical Regions, Tomato and Chili Peppers. What to do next? We do hope that we can establish a network with Tsukuba Plant Innovation Research Center and World Vegetable, and searching for research grants and develop of the Chaptex function, such as developing standard operating procedure and open public services for the clients. That is all our presentation. Thank you very much for your kind attention.
Thank you very much, Dr. Nono, for your report. Ladies and gentlemen, diving more into the research activity progress that has been given, we will continue to the question and answer session. I would like to invite every one of you, both who have attended offline and who have attended online, in this event who is willing to ask some question or give some feedback. You may raise your hand if you want to give question, maybe from the Zoom meetings. Or maybe in this room, if you have, please proceed.
Thank you, moderator. Maybe this is the discussion and maybe some advice that we can discuss with Bu Wulan and Pak Nono. Maybe first of all, thank you very much for the research, Bu Wulan. We see that your research result, that can maybe the projection, add some projection of the 2 to 3 years ahead, because we see that the policy in our country is it's unpredictable, especially shifting from last president to president in this year. This is very challenging for us. Maybe it can, in your research, it can add some projection of the, especially tomato and chili, because tomato and chili, we know that impact for our inflation, especially in price inflation. That is can some projection to our that commodity, and maybe we can learn from Japan how they manage the commodity inflation of that price commodity, because we see that Japan can handle that inflation product, and Pak Nono, maybe that's your objective or your Chaptex. It's about 2 new varieties that Pak Dekan is also mentioned that, because when and how to build the new varieties, both chili or the tomato, and also about the, oh, maybe I forget that. Next question, maybe that is one discussion, because Universitas Padjadjaran is push us, maybe Pak Nono now is the commercialization of our product research. It's maybe our challenge too. Thank you very much.
All right, thank you very much, Dr. Trida, for your feedback and input. Maybe from Professor Wulan or Dr. Nono, is there something you want to give feedback first? Enough? Please proceed, Professor Wulan.
Pak Trida, thank you very much for your suggestions. You suggested, if I can remember correctly, you suggested 2 projections for the tomato and chili. Let me first share my screen, especially about the calculations. Yeah, if we can see from here, yeah, we learn from the champion farmers here that we will say this is the feasibility study of the tomato and chili farms. And we can see here for the tomato, yeah, we can see in 1 hectare, the farmer can benefit a lot, yeah. So in 1 hectare, the farmer can produce, the productivity is around 28 ton per hectare. And then they can earn about 114 millions in terms of the rupiahs. And the profit that they can gain is around 52 million rupiahs, yeah, which is very nice, yeah, for one planting seasons, yeah. And as you mentioned before about the inflations, yeah, I agree a lot with you. When we can see from the calculation here, actually, yeah, the profit is very high here. So, but the challenge is when the harvesting comes, the price will decrease, yeah. So, because so many farmers grow the chili because of the profits that they can get, yeah. But then, when they grow it together, so we can imagine the harvest as well, yeah. So the price will decrease. Yeah. But when there is a lack of the chili, the price goes up a lot, yeah. Especially not only when the harvesting time, but also in the some events in Indonesia. For instance, when the Lebaran comes, Ramadan events, and also the New Year or the Christmas, yeah, events, the price goes up a lot, yeah. So in this case, yeah, we can see in the village, usually we can see very, very nice cars, yeah. Yeah, like Pajero and so on, yeah, they can buy, the farmers can buy, yeah. That's indeed contributes to the inflations, yeah, in Indonesia, especially for the chili. But yeah, I can see about your suggestions of the projections. Yes, within these 2 or 3 years, we hope, especially because this is only the original chili and the tomato. But then, because this project will produce some special products, yeah, they are the heat tolerance tomato and also the anthracnose chili, tolerance chili, that we expect that the economic analysis will also have the similar calculations we did in the original ones. Yeah. Thank you so much.
Thank you, Professor Wulan. Maybe Dr. Nono, you may proceed with your explanation.
Thank you very much, Dr. Trida, as Vice Dean for Academics, Cooperation, and Students. The question is about when our variety to be commercialized. Yeah, that is a good question, but the answer is not so easy. Because for developing one cultivars, let's say tomato or chili pepper, it might take around 5 or maybe more, 5 or maybe 10 years. But some also takes more than 10 years, around 12 and also 15 years. We do hope in our project it will take more 2 or 3 years. 3 years, yeah. Yeah, hopefully. Because currently we are in the step of creating variability, and some we did by a mutation, EMS mutation, and also we did that for genome editing. After that, we select the best one, so still takes time for the selecting the best one, and also we will check what we call the check the performance, let's say for yield and also the character that they have. For instance, in our project is the tomato tolerant to heat or high temperature, and also chili pepper resistant to anthracnose. So hopefully it will maybe 2 or 3 years more. Yeah, that's my answer. Maybe Dr. Farida or Dr. Chita will add some answer, explanation. Please.
Is there any additional from Professor Wulan or?
Thank you very much for the opportunity to add more information. We tried our best to to get a new variety, at least the prospective genotype in 3 years. But for a new variety, I think we need more than 3 years because we should do more evaluation in the 4th years. But at the end of this project, I do believe that at least we have prospective genotype for the new variety. And one of the best way to get the new variety in a shorter time. That's why in tomato we try to use molecular marker methods for the selection. So, for example, in Nil's population, if we do just conventional or classical breeding, then it will take more than 6 back crossings to get a good progeny. But by using the molecular marker, hopefully in the 3rd back cross, we can do the selection and we can succeed to obtain the prospective genotypes. That's why then we decided to use the molecular marker as one of the methods to shorten the breeding cycle. Thank you very much.
All right, thank you, Dr. Farida. Is there any more explanation, additionals? Enough? Okay, thank you. Maybe we have another question or feedback from Zoom meetings? Or from here, maybe another feedback? Please proceed. Yes, new questions. Yeah.
Jitskata from JECA Indonesia office. Thank you for your comprehensive presentation. So regarding, I think my question is mostly related to the economic part of this, or application of the output, like after developing these varieties. So first of all, do you have any observation among the land use, among the horticulture clubs in land use, basically the competition among the horticulture clubs? Because I believe that due to the free lunch program, the horticulture products is in more demand, and I thought maybe farmers, basically farmers, the preference in production may be changing. So do you have any observation if there is any, change in, so basically what to say, farmers' preference to continue producing chilies and tomatoes? So first, this is first question. And the other side, like if you look at the Captex, do you have any like competitor as a Captech? You have any, the universities or private sectors that is being, under the same practice or same research with as Captech? So there's 2 questions. Thank you.
Thank you, Mr. Jitsikata, for your question. Maybe from Mr. and Mrs. Researcher for the answer.
Oh, yes. Thank you so much for the questions. If I can understand correctly about your questions, yeah, regarding the, yeah, what I can understand is about the consumers' preference, yeah, especially about the demand. Oh, sorry. Yeah. Continue. Okay. Yeah, regarding the demand of the chili and tomato for our consumer, yeah.
Sorry, I was a bit, mixed up, but the consumer, not consumer, but the farmer's preference to produce chili. So is there any change in the the producer's preference in producing chili, continuing producing chili in the, in this this climate and also, the social context?
All right, thank you. So far, within these 3 years, when we visited farmers to interview, yeah, they are very happy to grow the chili, especially chili, yeah, because we know the inflation exists in Indonesia. But as I mentioned before, when they harvest, they can gain a lot of money from the chili. And for instance, in Garut district, what I can heard that when they harvest and they sell the chili, they can buy the very, very nice new car, like a Pajero here, yeah. And then it shows us that the potential for the chili to grow for the farmers, yeah, especially for the farmers in the productions area, still high, yeah. And then the second one is because the habit, the culture of Indonesians that cannot eat without sambal, for instance, yeah. So chili always needed in Indonesia. But yeah, I also can understand when we go abroad, like in Japan or in Europe, they only eat a very, very small size of chili. Because, yeah, you know, it again about the culture, but in Indonesia, I would say that the potential to grow chili is still there because of the demand also still high, yeah. Thank you.
Thank you, Professor Wulan, for your answer. Maybe is there any more additional answer from the researcher? Please proceed.
Thank you very much, Mr. Jitsukata, for your question. Your question is there any competitor for Chaptex? As far as we concern, until now there is no institution like the Chaptec. It's focusing on molecular breeding for chili and tomato, and maybe there is some of similar institution. Maybe it looks like similar, such as maybe in ITB there is ilmu hayati, biological sciences, related sciences, which focusing on biological sciences maybe. But until now there is no competitors. I mean, exactly the same like Chaptec. Thank you very much.
Thank you, Dr. Nono, for your answer. Maybe is that enough or any follow-up question? Okay, thank you very much, Mr. Jitsukata. And maybe we have another more question or feedback from this room. Is that enough? From the Zoom meetings? Maybe I will count down in 3 seconds. 3, 2, 1. Okay. If there's no more question or feedbacks, the last question we had just now signifies the end of the question and answer session. Thank you very much for your input and participation. Ladies and gentlemen, if the strength of an organization lies in its people, then today, Satraps Universitas Pajajaran team is gaining significant talent and expertise. We are exceptionally proud to introduce 6 brilliant individuals who are ready to dedicate their knowledge and energy to our research projects in agricultural innovation and sustainable development. We will call their names one by one. I would like to invite all of the audience to please prepare the warmest round of applause for each of our new members. The first new member, please welcome Ms. Putri Fajar Hairul Ramadhani. Next, Ms. Ataya Jamila. Ms. Nurul Fitri Hanifah. Mr. Fitriansyah Putra. Ms. Amanda Alia Sofian. And the last one is Ms. Lilian Rizky. Ladies and gentlemen, these are the 6 newest official members of the Satraps Universitas Padjadjaran team. We believe that with their combined passion and strong academic backgrounds in agriculture, these 6 new members will help us achieve the research breakthrough we aim for. Let's give them one more final massive round of applause. Thank you very much. And Excellencies, distinguished guests, ladies and gentlemen, we have finally arrived at the end of today's meeting. Joint Coordinating Committee Satraps Universitas Padjadjaran 2025. It was a really wonderful occasion we had. We, on behalf of the committee, would like to thank you for your participation and also enthusiasm. We also do apologize for any mistakes during this agenda. I am Risma signing out. Good afternoon and have a great day. Ladies and gentlemen, after this, let us take a quick photo session first. And we kindly invite all the participants to proceed directly to the main lobby of Rectorate Building because we will take the photo session there. Thank you very much.